Animal sex is far stranger than most nature documentaries ever show. Beneath the familiar surface lies a hidden world of stolen sperm, detachable organs, lethal mating, parasitic partners, and bodies shaped by conflict. This iceberg descends through one of the most bizarre corners of evolution, from well-known curiosities to reproductive systems so grotesque, intimate, and macabre that they barely seem real. In cyngnathids, pregnancy moved to the male. After courtship, the female deposits her eggs onto or into a specialized brooding area, and the father carries them until birth. In the simplest forms, this is external protection. In the most advanced, especially seahorses, it becomes a sealed brood pouch with blood vessels, controlled fluids, gas exchange, waste removal, and even placenta-like nutrient transfer. This is why male pregnancy in these fishes is more than parental care, it is a true physiological incubation system. It also creates an unusual evolutionary pressure. Because brood space is limited, males can become the selective sex, while pregnancy itself requires immune adjustments so the father can tolerate developing embryos without losing defense against infection. Among clownfish, sex is part of the social ladder. These anemone-dwelling reef fish live in small groups ruled by size. The largest individual is the breeding female, the next is the breeding male, and the smaller fish remain non-breeders. When that female disappears, the system does not collapse. The dominant male begins a transition into a female, and the next subordinate rises in rank to become the new breeding male. This is protandry, functioning first as male, then later as female. In biological terms, the change is real and deep. Male and non-breeding clownfish carry an ovotestis, and during transition the testicular part regresses while ovarian tissue expands. Research points to socially triggered neuroendocrine shifts, with aromatase, the enzyme tied to estrogen production, playing a central role. Many races live in strict social hierarchies, and in several species the largest dominant female can become the new male when the resident male disappears. On coral reefs, this shift can happen with remarkable speed. In the bluehead wrasse, behavior changes first, the future male becomes more aggressive, defends territory, and begins courtship displays. The body follows after. Ovarian tissue regresses, testicular tissue develops, coloration changes, and the hormonal balance is reorganized. This is protogynous sex change, a system shaped by size and social control. When large size brings the greatest reproductive advantage to males, evolution favors a life that begins as female and ends, for some individuals, as male. For bonobos, sex extends far beyond reproduction. These apes of the Congo Basin use sexual contact as part of everyday social regulation, especially in female-female interactions. Genitogenital rubbing appears around feeding, during tense moments, and in the integration of newly arrived females. Its function is measurable. Studies show that partners often stay close after these interactions, that female urinary oxytocin can rise afterward, and that dyads with more frequent sexual contact are also more likely to cooperate in coalitions. In evolutionary terms, this behavior helps manage friction, reinforce alliances, and support the female bonds that structure bonobo society. In this species, sex often works less as a mating event than as a social instrument. Barnacles look inert, almost plant-like, yet they are crustaceans locked into one place for adult life. That fixed existence creates a mechanical problem. Mating still requires physical contact. In most species, the answer is an extremely extendable muscular penis, used to reach nearby partners while the animal remains cemented to rock or shell. For their size, barnacles have the longest penis known in the animal kingdom, in some cases approaching 8 times body length. The advantage is simple geometry. A longer organ increases the area an individual can search for mates, while local wave energy can reshape that anatomy from season to season or from shore to shore. In barnacles, the giant penis is less a spectacle than an engineering solution to life without movement. Among mammals, echidnas belong to a very old lineage, and their reproductive anatomy follows its own design. In the short-beaked echidna, the penis ends in 4 rosette-like glands, yet only 2 are used during a single erection. The active pair can alternate from one mating to the next. Studies have also shown that echidna sperm move in bundles, a feature thought to improve performance under strong sperm competition. Taken together, this points to a highly specialized system shaped by breeding pressure rather than ornament, rapid copulation, repeated mating, and efficient sperm delivery in a species where several males may compete for the same female. In echidnas, the 4-headed penis is less a curiosity than a precise evolutionary solution. Among stiff-tailed ducks, especially the Argentine lake duck, the male organ reached an extreme form, a long corkscrew-shaped phallus, and in waterfowl more broadly its surface can carry ridges and spines that seem to help it maintain contact during rapid eversion. This anatomy makes sense in the context of sexual conflict. In ducks, forced copulations are common enough that penis length and complexity correlate with that pressure across species. Females, in turn, evolved complex vaginal tracts with spirals and blind pouches that can hinder full penetration unless they cooperate. In stiff-tailed ducks, the twisted and spiny penis is therefore best understood as one side of an evolutionary arms race, where access, resistance, and fertilization are all shaped by anatomy. In ducks, the conflict between male and female reproduction became visible in anatomy itself. Unlike most birds, many waterfowl males possess an intromittent phallus, sometimes long, spiral-shaped, and rapidly everted. Across species, that complexity tends to increase where forced copulations are more common. Females evolved the counter-design, vaginal spirals that turn in the opposite direction, along with blind pouches that can divert the penis away from the path to fertilization. Experiments on duck genital mechanics support the same conclusion. Full eversion works easily in a straight or matching spiral, but becomes far less effective in shapes that mimic the female tract. In other words, duck reproduction is shaped by an evolutionary arms race, where male persistence and female control are written into the body. In dragonflies and damselflies, mating includes a phase that looks less like courtship than biological interference. Before a male places his own sperm, he can use specialized genital structures to dislodge or reposition sperm left by previous partners inside the female's storage organs. In some species, this works through direct removal, in others, by pushing rival sperm away from the most effective site for fertilization, or by triggering the female to eject part of it. The logic is simple: eggs laid soon after mating are more likely to be fertilized by the last male in line. That pressure shaped an entire set of reproductive tools, turning odonate copulation into a contest over sperm already stored in the body. In several garter snakes, especially the red-sided garter snake, reproduction begins in a dense, moving knot of bodies. After winter, females emerge from communal dens and release pheromones that can attract dozens of males within minutes. The result is a mating ball, a mass of intertwined snakes, all competing to reach a single receptive female first. This behavior is driven by timing as much as by biology. Females remain fertile for only a short window after emergence, while males gather in advance around the dens, ready to mate immediately. What looks chaotic is actually a concentrated form of scramble competition, shaped by chemical signals, seasonal urgency, and intense rivalry between males. In red-sided garter snakes, a mating ball does not always form around a female. A small fraction of males release skin pheromones close enough to the female's signal that other males begin full courtship around them. These individuals became known as she-males in the older literature, males that chemically imitate a female while remaining anatomically male. The mechanism is grounded in pheromone biology. In this species, sex recognition depends heavily on methylketone blends in the skin, and experiments have shown that estrogen can shift male pheromone production toward a female-like profile. In many land snails, especially dart-bearing helicids such as the garden snail, courtship includes a shot. These animals are simultaneous hermaphrodites, so each partner can act as both sperm donor and recipient during the same encounter. Before copulation, one or both may drive a hard calcareous or chitinous dart through the partner's body wall. The dart itself carries no sperm. Its role is chemical. Mucus coating the dart delivers bioactive compounds that alter the recipient's reproductive tract, helping more of the shooter's sperm reach storage organs instead of being diverted toward digestion. The love dart is therefore less a romantic token than a tool for shifting paternity in a competitive mating system. In dolphins, the female reproductive tract is far more elaborate than a simple canal. Studies on bottlenose dolphins and other cetaceans show deep vaginal folds, side pockets, and asymmetric shapes that vary across species and even between individuals. Mechanical work suggests those folds can absorb and redirect copulatory forces, while comparative studies indicate they may also help reduce seawater intrusion into the tract, a serious issue for sperm function in an aquatic mammal. Their role likely extends beyond protection. Imaging and anatomical modeling suggest that female structure can influence how far the penis travels and where sperm is ultimately delivered. In dolphins, mating is shaped not only by behavior at the surface, but by an internal landscape that may itself be under sexual selection. In bed bugs, mating bypasses the genital tract entirely. In Cymex lectularius, the male pierces the female's abdominal wall with a specialized intromittent organ and injects sperm directly into her body cavity, a process known as traumatic insemination. The sperm then travel internally toward the reproductive organs. For the female, this is costly. Repeated matings create wounds, raise infection risk, and reduce survival and reproductive output. Evolution answered with a counterstructure. The spermolage, a specialized region that receives the puncture, helps limit damage, and plays an important role in immune defense. Studies even show that females can anticipate mating and increase local immune activity in advance. In bed bugs, sex is shaped by conflict, injury, and counteradaptation. In the tiny flatworms of the genus Macrostomum, mating can bypass the usual route entirely. These animals are transparent, free-living, and simultaneous hermaphrodites, meaning each individual carries both male and female function at the same time. In species such as Macrostomum hystrix, the copulatory organ ends in a needle-like stylet that pierces the partner's body wall and injects sperm directly into the tissues. The sperm then migrate through the body toward the eggs. This is hypodermic insemination, and in evolutionary terms it shifts control toward the sperm donor. Comparative work across the genus shows that this strategy evolved repeatedly, together with a distinctive suite of traits: sharp stylets, simplified sperm, and reduced structures linked to reciprocal mating. What looks like a crude shortcut is actually a stable reproductive system shaped by sexual conflict, repeated donor advantage, and the unusual biology of animals that are both sexes at once. In some marine polyclad flatworms, mating begins as a duel. Species such as Pseudoceros bifurcus are simultaneous hermaphrodites, so each individual carries both male and female function at the same time. When two partners meet, both try to inseminate while avoiding being inseminated themselves, striking with an everted, syringe-like penis in a behavior known as penis fencing. The logic is evolutionary rather than theatrical. Donating sperm is usually the cheaper role, while receiving it can lead to the heavier investment of producing and carrying fertilized eggs. What follows is a brief contest over reproductive burden, with sperm injected through the body wall and the outcome deciding who pays more for the mating event. Scyphopteron are small marine sea slugs, and in some species courtship begins with a brief assault. In Scyphopteron quadrispinosum, each individual is a simultaneous hermaphrodite, carrying both male and female function at once. When two partners meet, they first try to stab each other with a sharp stylet carried on one branch of a bipartite penis. That first strike injects prostate fluid through the body wall before ordinary insemination begins through the other branch. The sequence matters. Research suggests that stabbing first can push the interaction toward unilateral mating, allowing the successful individual to act as sperm donor while shifting more of the reproductive burden onto the partner. What looks bizarre is, in evolutionary terms, a conflict over sexual roles. In these slugs, mating starts as a contest between two bodies equipped for both sexes, each trying to secure the cheaper role first. On the surface of ponds and slow pools in East Asia, Geras gracilicornis lives like other water striders, balanced on surface tension, alert to the smallest vibration. Its mating system, however, is unusually coercive. Females evolved a genital shield that makes forced intromission difficult, so mounted males use a different tactic. They tap the water with their legs, sending ripples that attract underwater predators, especially backswimmers. Experiments showed that these signals raise the danger mainly for the female beneath the male, and females respond by exposing their genital opening sooner, allowing copulation to begin faster. In this species, courtship becomes intimidation, a sexual conflict in which a predator is turned into leverage. In honey bees, the males are drones, and their adult biology is built around a single event. They leave the hive to join aerial drone congregation areas, where virgin queens arrive for brief nuptial flights and mate with several males in succession. A successful drone pays for that moment with his life. During copulation, the endophilus everts with great force, semen is transferred, and part of the genital apparatus is torn away and left in the queen as a mating sign. The male then falls backward, fatally damaged. In evolutionary terms, This is terminal investment, one irreversible mating attempt, traded for a chance to place sperm in a queen who will store it for years of egg production. In several orb-weaving spiders, especially nephalenges and related nephilids, mating can end with the male sacrificing part of his own genital apparatus. Male spiders transfer sperm with modified pedipalps, and during or just after copulation one of those organs may break off or be severed, remaining inside the female as a genital plug. That fragment can obstruct later males and reduce sperm competition in species where females may mate repeatedly. The story goes further. Studies on these so-called eunuch males show that, after losing a palp, some guard females more effectively and can even gain endurance, suggesting that genital self-amputation is an evolved paternity strategy rather than accidental damage. Praying mantises are ambush predators, and in several species the male enters courtship as both mate and potential prey. Sexual cannibalism can happen before, during, or just after copulation, but in mantids it is best understood as a conflict between female nutrition and male survival. Work on the false garden mantis, Pseudomantis albofimbriata, showed that females in poorer condition were more likely to attack and consume males, and those cannibalistic females later produced heavier egg cases. Males, in turn, evolved caution rather than display. Many approach slowly, mount from behind, or attempt to mate while the female is occupied with feeding. In mantises, sexual cannibalism is less a grotesque accident than a reproductive system shaped by hunger, risk, and selection on both sexes. In a few orb-weaving spiders, mating can end with the male permanently damaging the female's external genitalia. Species such as Lorinia jascovi and Cyclosa argentioalba carry a small structure called a scape, used for genital coupling during copulation. After sperm transfer, the male can break that structure off with his pedipalp. The result is mechanical rather than symbolic. Later males struggle to couple properly, and the female's chance to remate drops sharply. Micro-CT studies and comparative work point to the same conclusion. This is a paternity protection strategy, evolved in response to sperm competition. More recent work also supports a deeper interpretation. Females can gain from multiple mating, so this mutilation is best understood as sexual conflict written directly into the reproductive anatomy. Spotted hyenas are among the few large carnivores where females dominate social life, and their anatomy reflects that exceptional biology. The female carries an enlarged clitoris, often called a pseudopenis, along with fused labia that form a pseudoscrotum. She urinates, mates, and gives birth through the same long urogenital canal. Developmental work shows that this extreme structure arises through unusual fetal pathways, with prenatal androgens involved, though the anatomy is not explained by hormones alone. The cost appears at birth. In first pregnancies, the opening must tear to let the cub pass, and studies on captive animals recorded stillbirth in roughly 60% of first births because the cub can remain trapped too long in that elongated canal. In spotted hyenas, reproductive control and reproductive risk are built into the same organ. Far below the sunlit ocean, serratioid anglerfishes live in a world where meeting a partner can be a matter of luck. The female is the hunter, carrying the lure, the jaws, and the full adult body. The male is tiny, with enlarged eyes and nostrils built mainly to find her. In several species, that search ends with a bite that becomes a merger. His tissues fuse with hers, their blood systems connect, and the male is reduced to a permanent sperm-producing organ nourished by the female's body. This is sexual parasitism, an extreme solution to mate scarcity in the deep sea. Genomic studies add an even stranger layer, showing that permanently attaching species lost key parts of adaptive immunity, making this living fusion biologically possible. Antechinus are small carnivorous Australian marsupials, and in several species the males are built for a single breeding season. For one short reproductive window, they roam, fight, and mate with extreme intensity, with copulations that can last many hours. Then the collapse begins. Rising androgens, stress hormone dysregulation, and intense use of body reserves push males into protein catabolism, immune failure, and synchronized death soon after mating, while females survive to gestate and raise the young. This is semelparity, one of the rarest reproductive strategies in mammals. The strongest explanation is ruthless efficiency. When the mating season is extremely short, sperm competition intensifies, and selection favors males that spend almost everything on one final reproductive effort. Argonauts are pelagic octopuses, often called paper nautiluses, and their mating system follows the same strange logic as life in the open sea. In this group, the female is enormous compared with the male, sometimes by an extreme margin. When a male reaches maturity, one specialized arm, the hectocotylus, carries sperm, enters the female during mating, then detaches and remains behind. For a long time, naturalists even described that severed arm as if it were a separate parasitic creature. In evolutionary terms, this system fits a lineage marked by extreme sexual dimorphism and rare encounters in the vast pelagic world. In Argonauts, the male does not simply mate. He leaves part of his body inside the female. We are now deeper in the iceberg, and oostacks are deep-sea annelid worms that live on the bones of dead vertebrates, especially whale falls. The female is the visible animal, a bone-boring worm anchored by root-like tissues and sustained by symbiotic bacteria. The males follow a very different path. They remain microscopic, larva-like, and gather inside the gelatinous tube that surrounds the female, sometimes in large numbers. From there, they function mainly as sperm producers. Research suggests that males and females come from the same larval pool, with settlement playing a major role in their fate. Larvae that reach bone develop into females, while those that settle on a female become dwarf males. In oostacks, mate scarcity in the deep sea is solved by turning the female herself into a permanent refuge for males. In the dry caves of Brazil, Neotrogla lives in a world where food is scarce and mating comes with nutritional rewards. These tiny bark lice feed in nutrient-poor conditions, and males transfer sperm inside large seminal gifts rich in resources. In this lineage, that economy appears to have reversed the usual balance of sexual competition. The female carries an elaborate intromittent organ, the gynosum, which penetrates the male's genital chamber, anchors itself with spines, and allows copulation to continue for astonishing lengths of time, sometimes for more than 40 hours. Research suggests that this anatomy evolved because the male's ejaculate is valuable food as well as gametes. In other words, the female is competing for access to a resource, and her penis is the tool that secures it. Chromadoris reticulata is a brightly colored reef nudibranch, a shell SC slug and a simultaneous hermaphrodite, so each partner can donate and receive sperm in the same encounter. What made this species famous is what happens after mating. Its penis is armed with backward-pointing spines, and the animal sheds the used tip after copulation. Within about a day, a new functional portion is exposed from a tightly coiled internal reserve, allowing another mating soon after. Later studies showed that those spines can also pull out sperm already stored from previous partners. In evolutionary terms, this is a highly specialized answer to sperm competition: transfer sperm, scrape away rivals, then discard the tool that just did the job. Banana slugs, the large forest floor slugs of the genus Areolomax, are simultaneous hermaphrodites, and in some species the penis is so elongated that it can exceed the animal's own body length. During mating, both partners can exchange sperm at the same time, but the process sometimes ends badly. If the penis remains trapped after copulation, one slug may chew through it, or both may do so, in a behavior called apophylation. The trigger appears mechanical, yet the evolutionary consequences may matter just as much. The injured slug can still reproduce in the female role, while its future male function is greatly reduced, which may also lower later sperm competition. Harpactia sadistica is a small disterid spider from Israel, and its mating system departs sharply from the usual spider pattern. Instead of placing sperm through the female genital opening, the male uses needle-like pedipalps to pierce the female's abdominal wall and inject sperm directly into her body cavity. During a single mating, he can puncture her several times. The female's normal sperm storage organs are greatly reduced, and fertilization appears to take place internally, with eggs already developing before they are laid. The leading explanation is sperm competition. By bypassing the usual storage route, the first male may gain a stronger paternity advantage and make later sperm removal far more difficult. Melitobia are parasitoid wasps that develop on the larvae and pupae of solitary bees and wasps. Inside that closed nursery, the sex ratio is pushed heavily toward females, and the few males that emerge are blind, short-winged or flightless, and trapped on the natal patch. Their entire reproductive world is immediate and familial, sisters as mates, brothers as rivals. What follows is brutal. Males attack each other with their mandibles, maiming or killing competitors before newly emerged females become available, then switch to pheromone-guided courtship once a sister is ready to mate. The result looks like sexual civil war, but its logic is precise. In a brood where mating happens locally and quickly, females gain by producing mostly daughters, while each male gains by removing the few rivals standing between him and the next female. Among widow spiders, especially the Australian redback, mating can end in deliberate self-sacrifice. The male is tiny beside the female, and a successful encounter is rare enough that every copulation carries enormous value. After achieving genital coupling, he performs a somersault that places his abdomen directly over the female's mouthparts, inviting cannibalism while sperm transfer continues. This behavior looks suicidal because it is. Yet experiments showed that cannibalized males can prolong copulation and gain a stronger share of paternity, while later mating opportunities are scarce because mate search is dangerous and females are widely spaced. In evolutionary terms, this is terminal investment. The male turns his own body into part of the reproductive strategy, trading survival for fertilization success. Mallards are familiar dabbling ducks, yet their mating system can be intensely coercive, especially once pair bonds begin to loosen in spring. In waterfowl, forced extrapair copulations are a well-documented part of sexual conflict, with males chasing, pinning, and mounting resistant birds. Against that background, the most famous necrophilia record in birds came from Rotterdam, where a male mallard continued copulating for about 75 minutes with a freshly killed male that had just struck a glass facade. The original interpretation was an attempted rape flight that ended in a fatal collision, then continued because the pursuit and mounting response had already been triggered. Scientifically, this is best understood as a rare spillover of extreme reproductive drive, not as an ordinary feature of mallard social life. Fig wasps spend almost their entire sexual life inside a fig, which is actually a closed inflorescence known as a syconium. Their larvae develop within individual flowers, sealed from the outside world. In many pollinating species, the wingless males emerge first, move through the fig cavity, and mate with females that are very often their sisters, sometimes before those females have even fully left their natal chambers. After that, the males usually chew exit tunnels through the fig wall, allowing the newly mated females to leave, collect pollen, and search for another figure. Most males never leave at all. The logic is evolutionary rather than accidental. This is a system shaped by extreme local mate competition, where mates are concentrated in one enclosed nursery, brothers compete with brothers, and female-biased sex ratios become the most efficient outcome. Among parasitoid wasps, Incarcia formosa offers one of the strangest reproductive systems. It is a minute wasp that develops inside whiteflies, and like many parasitoids it belongs to a haplodiploid lineage, where sex is normally tied to whether an egg is fertilized. In that ancestral system, unfertilized eggs produce haploid males, while fertilized eggs produce females. In Incarcia formosa, that rule can be hijacked by a bacterium. The symbiont Wolbachia induces female-producing parthenogenesis, effectively turning would-be sons into daughters and allowing reproduction without ordinary mating. A 2024 study showed that the bacterium carries a feminizing gene, called PIF, that interacts directly with the wasp's sex determination pathway. In evolutionary terms, this is more than virgin birth. It is a sexual system partly taken over by a microbe. For most of the growing season, an aphid colony expands without males at all. In many temperate species, spring and summer are dominated by live-bearing clonal females, and the system is so compressed that a mother often carries developing daughters and early granddaughters at the same time, the classic telescoping of generations. Then the season shifts. As nights grow longer, that photoperiodic signal pushes development toward sexual forms, males and egg-laying females. After mating, those females produce frost-resistant eggs that can survive winter and hatch the following spring. In aphids, sex works almost like a seasonal emergency protocol, triggered after layers of nested asexual generations, less for speed than for survival and genetic reshuffling. Aphids have an even stranger second layer to that seasonal switch. After long stretches of clonal reproduction, the sexual phase produces males in an unusual way, by losing one X chromosome. Females are XX, while males become X0 during a modified division, effectively created by subtraction rather than by fertilization alone. In some species, that discarded X appears to be lost at random, which makes male production part of the same emergency reproductive program triggered by autumn light cycles. So this second aphid point is not about a new animal at all, but about the hidden mechanics inside the sexual phase itself. First, sex returns as a seasonal backup system, then males appear through chromosome loss. Eisseria purchasei, the cottony cushion scale, is a sap-feeding insect and a serious plant pest, but its sexual system is far stranger than its appearance suggests. Most individuals are female-like hermaphrodites. Inside one body, the reproductive organs produce both eggs and sperm, and those sperm can fertilize the animal's own eggs through a shared duct system. In practice, a single individual can found a new population alone. Yet the system is not fully closed. Rare winged males still appear, and genetic studies show that outcrossing does happen from time to time. This makes Eisseria purchasei one of the clearest animal examples of androdiocy, a reproductive strategy that combines the colonizing power of selfing with the occasional return of genetic mixing. Bonellia viridis is a marine spoonworm, and its sex is decided after the larval stage by the environment it encounters. A larva that settles alone on the seafloor develops into a large female. A larva that lands on an adult female, especially on her long proboscis, follows a very different path. It is drawn into her body and becomes a minute dwarf male, living inside the female and functioning mainly as a sperm producer. Experimental work showed that this shift is triggered by chemical signals from the female, with the green pigment bonellin long implicated in that masculinizing effect. In Bonellia, sex is not fixed at birth. It is imposed by contact, turning a chance encounter into one of the strangest forms of environmental sex determination in the animal kingdom. At the bottom of this iceberg sits a dactylidium, a microscopic mite that turns one thrip's egg into an entire reproductive chamber. A single mature female feeds on that host egg, swells dramatically, and produces a brood of about 6 to 9 offspring, usually with only one male among them. Before any of those young leave her body, that brother mates with his sisters inside the mother. The fertilized daughters then tear their way out through her tissues, killing her as they emerge. In life history terms, this is an extreme compression of reproduction, one tiny host, almost no opportunity to search for mates, and a system that forces fertilization to happen immediately, inside the only shelter available. At the very bottom of this iceberg sits Acarophonax tribolii, a microscopic egg parasitic mite associated with flower beetles in stored grain. The mother settles on a host egg, feeds, and swells into a living chamber packed with offspring. Her brood is built with ruthless economy, many daughters, usually only a tiny number of sons. Those sons mature first, mate with their sisters inside the mother's body, and in this species the male can complete his entire life cycle there, dying before he ever emerges. The daughters then leave already inseminated and ready to repeat the process on another host egg. In evolutionary terms, this is local mate competition pushed to an extreme limit. When mating happens inside a single body, a male becomes useful for only one brief task, then disposable.